Thank you very much. It is a real pleasure to be able to share some of the work that we currently do at CERBA and give you kind of a broad overview how CERBA scientific team looks at biomarker. The title I chose for this presentation is "A Single Picture Does Not Say It All." And what I hope you will get at the end of this presentation is the fact that biomarkers have grown extremely complex in the past few years, but nonetheless are extremely exciting and hold a lot of potential in advancing clinical research. Just as a brief presentation, my name is Eitan Akirov. I am VP of Business Development and Global Lead for Biomarkers at CERBA Research. In this presentation, I would like to cover the following four main topics. The first one is really to answer the question on why biomarkers are critical for any clinical program that's considered bringing a therapeutic to market. The second question is, should we actually use biomarkers in clinical research, given the fact that, of course, they could be expensive and time-consuming. The third point was a case study for adopting a comprehensive approach for biomarker design. I will put special emphasis on solid tumors in oncology and cancer. And finally, I will give a bit of a flavor of the different approaches that are being used for clinical biomarker research at CERBA and elsewhere. So I want to spend no more than a few seconds about CERBA Research, just to give you a bit of an introduction about our company. We consider ourselves as a universal, if you will, one-stop shop, a service provider under the CERBA Research umbrella. We have four main types of services that we offer: clinical trial laboratory services, a dedicated team for IVD diagnostics validation, as well as development, translational research, and finally, the team that deals with biomarker and companion diagnostics, which I am part of. As part of the biomarkers offering that we have, we really try and cover pretty much all of the common areas and techniques that are being used in biomarker. Many of those are done in-house, some of them with collaborators. But all in all, from flow cytometry to IHC, genetic and genomics, molecular biology, metabolomics, and PK/PD, we really try and offer a single harmonized solution for any type of clinical research. We are global. We have presence in pretty much all continents, very strong presence in Europe and the United States, as well as in Asia Pacific, and one of the leading laboratory partners in South Africa. This really allows us to handle all the aspects of shipment logistics and assay harmonization between wet laboratories. All right, so I'm going to jump directly into the kind of the topic in hand, which is what is the current probability of success when one looks at clinical trials, and whether those type of probability of success can actually be influenced by the use of biomarkers. So the first point that comes to mind, and this is a review that was published in Nature Reviews Drug Discovery by Richard Harrison, is that oncology trials, as common as they are, they account for the vast majority of clinical trials in the market, really have the lowest success rate in all therapeutics areas. And this success rate, poor success rate, is really a trend that has been documented by him and others since 2007. And aside from a very minute and marginal improvement in success rate, unfortunately, there hasn't been major improvements in the ability of bringing new compounds or biologics into market. And finally, the main cause for failure, especially in oncology, is really the inability to show adequate efficacy. And this was, quite frankly, a surprise to me when I first saw this data, because one would expect that at least the transition from phase 2 to phase 3 would already give indication on whether a drug is efficacious or not efficacious in treating this specific tumor or specific cancer. And I think if you take all of these elements into account, it really paints a picture that requires better ways of improving the probability of success for clinical trials, specifically in oncology. So in a really beautiful paper published by Wong et al. From MIT back in 2019, they looked at what is the probability of success as you move throughout the clinical phase. I would call it process. Phase 1, phase 1 to 2, 2 to 3, 3 to approval, and overall approval. I summarized some of the data here in a graph, and I think what is evident right away is that the highest success rate is in phase 1. I think this is quite self-explanatory because in that case, efficacy plays a relatively low role. People normally focus on a small cohort of patients looking at the ability to demonstrate safety first and foremost. Once you move from phase 1 into phase 2 and phase 3, there is actually a detrimental effect on the probability of success as the levels go down. And finally, the blue bar that you can see, the dark blue bar that you can see in the graph, which represents oncology, more than 17,000 data points incorporated here, show that the overall approval rate for oncology is really a mere 3.4% at the end of this process. And this is quite tragic because about 26.5% of all attempts are doomed to fail. So the group at MIT decided to see whether biomarkers actually make a difference, and I think that is a main differentiator in this particular report. And the conclusion that they drew coming from this particular study was that indeed the incorporation of biomarkers, specifically in oncology, make a huge improvement at the level of success as you move throughout the clinical trial development plan. The red bars that appear in the graph show the success rate in phase 1 to 2 all the way to approval. Only about 10% of clinical trials used biomarkers, which is quite interesting. It means that this has not been adopted early on once those clinical trials were actually conducted. And finally, when you look at the impact of incorporating biomarkers in clinical trials, it is staggering to see that by including biomarkers in a clinical trial, one can drive success rate by more than 5-folds. And this, to me, is and should be for the scientific community a clear indication that biomarkers can make a difference when it comes to bringing a new drug to market. So what is a biomarker? And I think the best way to define it is to go to the agency that actually looks at the data at the end, the Food and Drug Administration, the FDA. And when you read the definition of biomarker, it is really all-encompassing. It covers pretty much any indicator that can look at biological or pathogenic processes as an individual is exposed to any type of intervention, including therapeutic interventions. This definition is great because it puts a very broad umbrella on what a biomarker is, but interestingly enough, things that a scientist may not necessarily define as a biomarker also fall within this category. Here we're looking at blood glucose, which has been around for more than a century now, blood pressure, which is more of a physiological type of measurement, and tumor size. And I think pretty much everybody on this conference agree that biomarkers definition has become a much more deep and much more broad when we're trying to look at ways to measure a response to any type of intervention. Okay, so this really leads to the next point, which is if we look at biomarkers, they actually span a very broad definition ranging from the way that they are used to the way that the matrices that are being collected as well as the techniques. When one looks at matrices, any type of investigator really, if possible, can and should tap all of the lists that are listed here, different bodily fluids, looking at tumor microenvironment by biopsies, looking at tumor macroenvironment by imaging, and looking at circulating cells, whether they are of tumor origin, immune origin, or just healthy cell as a comparator. When we look at the way that these biomarkers or these matrices can be used, again, it is a very broad list of uses for biomarkers. These include diagnosis, prognosis, monitoring of the disease, tumor evolution, if we're trying to track whether a tumor is becoming resistant to a specific type of therapy, patient stratification, which is key, because in that paper that I mentioned before from Wong, that was the determining factor for using biomarkers, measurement of drug safety, and of course, measurement of drug efficacy. When one looks at the type of biomarker techniques that can be used, the topic becomes even broader. We look here, I gave just a partial list of the type of areas that biomarkers could be looked at, from molecular to genetic histology, different metabolites, liquid biomarkers, as well as theoretical and in silico biomarkers for data analysis. And within brackets, I put a list of some of the most common techniques that are used to tap those. So when one really discusses biomarkers, it is really a very impressive, but nonetheless complex and comprehensive umbrella of a topic to cover. Okay, so I thought it would be quite useful to go ahead and maybe pay special attention into solid tumors, and within those solid tumors to look at the different ways that we can look at solid tumors with a strong focus on immunology, on the immune system. The immune system is key at not only protecting us from infectious agents such as bacteria and viruses, but it also plays a critical role in tumor surveillance and making sure that malignant cells are being eradicated before it's too late. Within the immune system, lymphocytes can actively penetrate the tumor. Those are known as TILs, or tumor-infiltrating lymphocytes. And when we analyze those lymphocytes, is a part of our routine activity that we actually do at CERBA, we look at a snapshot of what these cells tell us. So they tell us what type of cells are actually being infiltrating the tumors, what is the level of activation, what repertoire do they have. It allows us to look at changes in immune activation over time in cases that we have the opportunity to look at multiple biopsies over a duration of a study. It also helps us identify new therapeutic targets by characterizing the cells, as I'll show you in a second. And finally, if they are done early on, they can be successfully used also for initial patient stratification to make sure that the right therapy is being offered to the right patient. So this little cartoon here actually demonstrates the type of cells that could be found. For the interest of time, I will just mention that the majority of the cells that are measured normally are of the innate and the adaptive immune system, and those are the most active cells in tumor biology. So as a very simple and straightforward example, we decided to look at non-small cell lung cancers and NSCLC. At CERBA, we developed a robust multiplex IHC approach in order to look at multiple biomarkers of the disease. Many of our partners ask us why to use multiplex, and there are really four immediate reasons. Biopsy size is normally limited, and it's hard to find enough numbers of sections to do the analysis. People would like to see more and more biomarkers on a single slide because it gives so much additional information on co-staining and characterization of cells. It gives us the opportunity to do accurate phenotyping, and something that I think is really the highlight of IHC and any type of histology is that you get to see the morphology of the tissue, which gives you both spatial context and distance, something that other techniques such as flow cytometry, which I will cover next, cannot really do. So in this first example, I wanted to share with you this image of looking at myeloid-derived suppressor cells. Those come in two flavors, polymorphonuclear and myeloid, and you can see here the way that multiplexing has been applied to look at several different biomarkers, CD1, HLA, and so on, to characterize these two cell populations. The beauty of it is that we can actually not just characterize them, but also measure the distance from where the tumor tissue can actually be identified. Looking at another element, a very well-established element of immune suppression, our suppressor regulatory T cells. These are also known as Tregs, and these cells are normally identified by a classic T cell biomarker such as CD3 and CD4, as well as activation and suppression transcription factor FOXP3 and CD25. When looking at here again, there is a quadruple staining for these cells, showing them in close proximity to CD3, CD4 cells, again suggesting that the immune system has activated a suppression mechanism in this particular cancer case. So this was a kind of a very quick review of zooming at the tumor, but can we actually zoom out and look at cells that are in the periphery? I think that question is extremely important because biopsies are not always available. That's the first reason. And even if they are available, they still do not give us a more of a comprehensive view of the immune activation in cancer. When we look at peripheral lymphocytes, those are the ones that will be found in the blood, in the circulation. They also, very similarly to TIL, provide us a snapshot of immune cell types in the blood. They give us a good measure of how our cells are doing in terms of activation, energy, and generating memory responses. Once the therapeutic is applied, it's relatively easy by just drawing blood to look at what the impact is on immune activation. This is extremely important in the cases of checkpoint therapy as well as CAR T-cell therapy, and also important for identification of new therapeutic targets. Again, similar to what I mentioned for TILs, there's a clear ability to look at innate cells as well as adaptive immune cells, as well as characterizing malignancies as pharmacodynamics and receptor occupancy. All right, so the most, I think, used and well-established method for characterizing circulating lymphocyte is, of course, flow cytometry. This is a case example where we have applied flow cytometry, multiple gating, multiple staining in order to identify a panel of memory T cells. Without going into too much details, the process requires identifying live cells, as you can see here on the left panels. Once the live cells have been successfully identified, they then get stained for common T cell biomarkers, very similar to the one that I've showed you for IHC, such as CD3 and CD4, as well as some chemokine receptors that help the investigator identify memory T cells, and then zoom back on the frequency of central and peripheral memory T cells. You can probably appreciate the fact that this is a perfect example where you can correlate data using the peripheral lymphocytes and the TILs because the biomarkers that are being used are being shared by those two individual techniques. Another method of looking at circulating lymphocyte, which is exciting and emerging, is TCR-seq. TCR-seq uses sequencing in order to identify the variable region of the T-cell receptor. This is a beautiful example from Shea et al. From Nature Communication, a paper that was published earlier this year. They looked at the evolution of ALL and non-Hodgkin's lymphoma patients after they received engineered CAR T cells targeted against the CD19 receptor. This beautiful paper looked at the frequency of the most common, you can see here the most common clones that were actually generated. This is pre-injection, and then looking at the kinetics. And the thing that's most impressive and staggering is that every patient will have a different type of selection of T cells after they are introduced and engrafted into the host. And this really paints what we all know is that every patient is unique and the system is unique, but also highlights the fact that without biomarker, this would have been really a black box. Looking at another aspect of circulating lymphocytes and applying next-generation sequencing techniques, the group also looked at the expression of different genes. You can see that this list of genes really encompasses everything from HLA-related genes to T-cell activation, cytokine production, and so on, again demonstrating the point of evolution of these cells throughout the process of engraftment within the patients. And finally, and I think very importantly, this NGS technique also allowed the investigators to look at the expression of Ki67, a biomarker for T-cell proliferation. T-cell proliferation within recipients of engineered T-cells is critical, as well as the cytokine storm, which could be detrimental for the successful of the disease. And I think, again, the use of the intelligent use of biomarkers in this case really contributed a lot to the knowledge of the therapy in these patients. Finally, to look at another aspect of biomarkers, we all know that lymphocytes can exert their effects by cell-cell contact, but lymphocytes are also very active secretors of pro- and anti-inflammatory factors that can control the tumor environment, tumor growth, and tumor destruction, ultimately. These secreted soluble factors can be measured both in an autocrine, paracrine, or endocrine manner because they can be impactful on both the microenvironment and the macroenvironment of the tumor. So when one look at those, one can actually look at the soluble factors such as cytokine and chemokine secretion in the TILs, as I mentioned before, but also can look systemically to identify these factors as they are circulating in the body. Similarly to the IHC and the flow cytometry point I discussed earlier, the correlation between these two could be very informative and very helpful in understanding the activation state of the immune system. So how do we actually look at those? This topic is extremely broad, and for the interest of time, I'm just going to give you two examples on how we look at these, both a direct secretion from the cells as well as soluble factors that are secreted in the body or into a specific matrix. For direct secretion, one of the most common techniques are ELISpots. ELISpots is a technique, a well-established technique that exists for decades now, whereby normally T cells as well as B cells are activated against an antigen. They secrete a cytokine into the well, which then allows the investigator to enumerate the number of cytokine-producing cells, antigen-specific cells. In addition to that, an extension of the flow cytometry staining that I showed you before is the ability to look at intracellular cytokine staining. In this case, we're looking at CD8 cytotoxic T cells, showing a NUS population of TNF-alpha and interferon gamma positive cells. Again, a very important characterization of understanding the type of cytokine milieu that these cells actually produce. For looking at soluble factors that come from the circulation, we can always use serum and plasma, as well as cells that are stimulated in cell culture media, and then analyzed using immunoassays. One of our preferred methods in-house is use the MSD platform. This is an electric chemiluminescence system that allows the multiplexing of up to a dozen factors within a single well, which allows us to both get a very broad picture, but also to use very small amounts of sample in cases where the availability of serum and plasma are limiting, such as in pediatric studies. All right, so to sum things up, it is quite clear that as the years pass on, there is going to be a higher level of use of biomarkers. These biomarkers will become much more specialized, looking both at a cell phenotype, but also at biological pathways. The techniques that are being highlighted in this particular report from 2018 really calls the same techniques that I've tried to cover today, including IHC and flow, next generation sequencing, as well as molecular biology. And what I hope I was able to convey to you is that choosing a single snapshot or choosing a single technique in order to look at a specific biomarker or the specific condition is most likely not going to be enough. It's the application of these four main techniques that I try to cover today: flow cytometry, IHC, genetics, and molecular biology, that really paint a complete and an important picture of the status of immune activation in the case of immune-oncology and in all other clinical trials for that matter. So I wanted to thank you and give you my contact information. I am located in the United States, but as I mentioned to you before, our organization is global, and we would be always happy to help you with design and execution of your clinical trials. Thank you.